Okay, yeah, thank you very much. First I'd just like to thank everyone for participating today. It's amazing to see so many people here. I think we had close to or more than half of the attendees for the entire meeting actually here present today at Education Day. So I think this is an amazing sign. As you can tell, we worked very hard, and especially our speakers today, to include a lot of information that's more recent and modern. So probably not trying to cater maybe towards the newest person in the field, but actually talk about some of the complexities and hurdles and other aspects that we're still facing right now, so that maybe a lot of you will actually innovate in these areas. So a conceit that our committee had many months ago when we first started organizing this day was to actually maybe have a part of the session where we talk about some common misconceptions of EVs, maybe more focused on their physical characterization or some of their presumed functions. And then we kind of got scooped all day because all of our speakers were bringing up these amazing misconceptions and challenges and hurdles and complications. But I still think we have a few interesting tidbits that maybe weren't covered today, or some that I'll actually just talk about in a little bit more detail. Okay. So if you were to look at any of the recent EV reviews, you'll find something very common, and there's this picture that I hate, no offense to the artist. I'm gonna say many things that probably people that made these are in the room, and I'll just apologize up front. But if we believe these images, then it's pretty apparent that EVs are unilamellar. They each carry every single marker that we think are on them, and they're all quite homogeneous. Of course, as we all know, that's not the case, right? These are literally just the front page of Google results of searching extracellular vesicle and looking for images. And I think one theme of this talk and probably of the day is that our cartoons are too simplistic and too reductionist, and actually the real picture of EV heterogeneity is quite beautiful and useful, and probably we can do a better job of animating these when we're actually talking about EVs. So I really like to think about the more physical parameters of EVs. So I did my best on this cartoon schematic to try to draw things to scale. So this is roughly the size of a lipid, the thickness of a lipid bilayer, the size of a membrane protein, the size of a sterol, the size of a glycan, and then the relative sizes of the sort of quoted size distribution of smaller EVs. You'll often see 30 to 150 nanometer. We'll talk about how that's, of course, not true and it's complicated. But as you can see here, look at how small the intraluminal volume is of a 30 nanometer vesicle compared to a 150 nanometer vesicle. It is impossible that these things have the same biodistributions and the same functions and the same biomarkers or whatever, right? And you can do some pretty simple thought experiments where you consider the average size of a membrane protein, a few nanometers squared, and you can figure out the surface area of a typical vesicle, and you can just do some simple math. If a 30 nanometer vesicle was completely packed with membrane proteins, as much as it could be, meaning there's no lipids, you could hold about 100 single proteins. Of course, we know that the bulk material in the lipid membrane is lipids, so of course the real number is much lower. But now let's just consider a 150 nanometer diameter unilamellar vesicle. The same number of proteins is 2,500. So a huge discrepancy of more than an order of magnitude difference between these. You see that relative surface area is 25 times higher. The relative volume is 125 times higher because you scale with squared and cubed. Of course, there's a lot of historical work that show interesting stoichiometry, not based on thought experiments like this one, but actually measuring the sort of expected amount of microRNAs that and that's this dotted line here, that should be reached if there was enough that there was one copy per vesicle. And I would say this is already a quite outdated study that probably severely undercounted the number of EVs. So the situation is probably much worse than this, or better if you're a fan of EV heterogeneity. And you can see that even for the most abundant microRNAs, there is not enough to fit in every single copy. So of course, these things are highly heterogeneous, and each vesicle only carries a very tiny subset of each individual molecule that you're looking at. And this assumes that even amongst all of those, they're distributed evenly, which of course some particles are going to be enriched with certain ones more than others. So as we heard about today, there's just so much interesting work being done in digital analysis and single EV analysis where people are starting to consider the implications of this heterogeneity. Here's a nice study, actually it's not from 2014, it's from 2023, my bad, where they're actually doing single EV imaging and starting to try to work out some of these differences in stoichiometry. And what you see are these beautiful heat maps. So these are really distinct subpopulations that we see of co-localization of different membrane proteins on single EVs, depending on the capture, depending on the source. And just, from a bird's eye view, you can see that these things are quite heterogeneous. And this type of analysis might actually reveal that there are multiple types of subpopulations that actually can be distinguished by composition rather than size or other physical properties. There's other work in this area looking at the sort of classic tetraspanin, these three super reductionist tetraspanins that everyone is insistent on using, even though I remember coming to my very first ISEV in 2015, and it seemed like half of the speakers in that event were saying, oh, everyone's using these tetraspanins, why are we doing this? And here we are 10 years later doing this still. Needless to say, which tetraspanins are on different subpopulations, as we heard from Clotilde's talk this morning, can be indicative of biogenesis pathway, but it's highly variant on source. So you can see massive CD1 detection inside of some cancer cell EVs. In serum, there's very little. In PMSCs, there's more or less an even distribution. And there's a huge bias. If you're using tetraspanins to capture EVs and then looking for co-localized biomarkers, these are just some generic cancer ones, CD24, EpCAM, and HER2. It actually depends on which marker you capture if you're biasing yourself towards the individual markers. So you see CD24 in these patients were more correlated with CD81. So if you would have pulled down by CD63, you wouldn't have seen that at all and probably concluded that this patient didn't have any disease burden. HER2 seemed to be more evenly expressed across them. And that last bar, this fourth bar here, is for nonspecific capture of just biotinylated EVs through nonspecific capture of amines, and that was consistently the highest on all of them. So again, thinking about cartoons and sort of the pictures that we represent these, I think it's important to start drawing EVs like how we see them on our imaging techniques, and I'll show you some images, but of course we start to see people drawing these with heterogeneity in size, but also lamellarity is a massive thing. And of course we now know that there are lots of particles that actually aren't vesicles, but are sort of in between these sort of aggregates that we typically think of, solid particles like lipoprotein, ribonucleoprotein particles, which is an emerging field of RNA binding proteins in RNA that can be as big as EVs, as we typically think of EVs. And of course, overlaid on all of that is this complexity in molecular content. And we've heard today, and of course most of the literature is focused on the proteins, maybe the contained nucleic acids. Of course, we have recent evidence that nucleic acids are largely absorbed to the surface of the particle. You rarely see that drawn in cartoons. Metabolites and small molecules are largely ignored, other than maybe a footnote in the first paragraph of most papers saying that they're trafficked in EVs. The complexity of sterols and the influence on membrane rigidity is something that's just emerging. Glycans, in my opinion, are completely understudied in EVs, and these are widely appreciated in cancer biology to dictate tropism at the cellular level. And of course, as evidenced by a recent Nobel Prize in chemistry just to study these, they're actually difficult to study, and that's probably why it's lagging. But these are big opportunities to actually consider true heterogeneity and diversity of EVs. And we're starting to see much better images, if I can editorialize and say that this is better, because it actually is starting to appreciate that diversity of the different particle types which are emerging from cells. And then there's this, which was shown a couple times today, showing this picture that I sort of ragged on in slide one, showing that, this classic image of a beautiful homogeneous EV, which contains every class of biomolecule that's measured in the bulk. And then actually real EVs are probably messy. Many of them are probably empty. I think this picture goes a little too far. We certainly know that there's a large population of EVs that have multiple markers. And again, when you shrink the EV, why would the lipid bilayer shrink as well? These are things that annoy me in drawn literature, so I apologize if you're in the room, but I'm happy to rag to your face as well. And then the size, this is another persistent myth. So if you look at any papers, everyone's confused. If I look at my own papers from the past 10 years, I don't think I used the same size range once. So they're 40 to 150, they're maybe 30 to 100, maybe 50 to 500. Most people agree that the mode is 100, which now we know is not true. And there's lots of good evidence that a lot of that conventional wisdom are huge artifacts of the tools that we're using to study EVs, namely light scattering. I'll get into refractive index next. Some careful work by Edwin van der Poel and some colleagues have sort of started to stitch together different modalities of EV sizing. So cryo-EM, which is very good at counting the smaller ones. You're actually visualizing those and circling those, and then that's in blue. Then in black here you see the single particle flow cytometry, which at the time of this work had a cutoff of around 160 nanometer. And when you start to stitch those together, you actually see this really interesting, what I would consider the true distribution of sizes for EVs, which is this log linear plot, where you get a log decrease in concentration as your particle size scales linearly. And the vast majority of EVs by count, by surface area, and by volume are completely undetectable by most light scattering methods, maybe with the exception of some of the newer devices that are on the market, such as these sort of next generation of single particle flow cytometers, super resolution microscopes, and lots of the technologies that you heard about today, and of course, electron microscopy. Refractive index, again, in my opinion, this is all in my opinion, is completely understudied for EVs and the implications of what we're assuming when we do these type of models. So if you're not familiar, this is sort of a complex, and I mean complex in terms of using that as a mathematical definition, property of particles which is dependent on the light attenuation or the absorption of light by these particles, in addition to the angle of the refraction, the way that the light scatters out of this individual particle. What that leads to is these very funny curves. So this is the Mie scattering model, and because of this strange attenuation, you get these little resonances, these small peaks. And what you see here, looking at polystyrene versus extracellular vesicles, is that the amount that light actually gets scattered from those particles is first dependent on the diameter, but also dependent on the overall refractive index. So how the materials and biomolecules that comprise EVs are fundamentally interacting with the light that's passing through. And polystyrene, as compared to EVs, are of course more refractive. And so at equivalent size, they scatter a lot more light. Smaller particles, of course, scatter less light, but at the same diameter, the EVs scatter a lot less light. And we've known this for many, many years, and this has huge implications on sort of the optics of scattering detection. So here's a cross-section of how a flow cytometer is picking offside scattering. You see that the angles can be precisely determined, and all of this can be mathematically modeled and fit using standards, and this is sort of that my flow site framework developed by Josh Welsh and a lot of collaborators at that time, which is being widely used. The plot thickens even more because the situation is even more complex, because EVs, of course, are not homogeneous. So you have where you have this single sort of refraction of light at the surface, and then at the other surface. In EVs, there's many, many surfaces. Light is interacting with all of the molecules that are comprising the EVs, including the inside and outside of the lipid bilayers on both sides and all the molecules in between. So even on our most advanced models currently that are trying to turn side scattering and determine size, they're not considering that EVs have a natural diversity in refractive index, which is related to their materials. So a lot of these you'll see in the NanoView system, in all flow cytometry modeling, are turning scattering into size, and they're all assuming that EVs have a static refractive index. So all EVs, big and small, they say are 1.4, or whatever the number that you might be able to float. In fact, of course, we know that's not true, and you could easily just look at cryo-EM images to actually tell that. You can use the density of the particles as a surrogate for their refractive index. If you look at these two purple particles, these are pretty much the exact same size in their nominal diameter. One is quite dense, and one is quite empty. These are going to have completely different refractive indices. So if you assume a constant refractive index and turn that scattering into size, you'll assume that these two particles had different sizes. Similarly, you can see that the internal contents of EVs is not homogeneous, even when there is a high electron density. Multilamellarity plays a big role in that scattering. You could see that some particles clearly have a weird distribution spatially inside of them based on the content. So they're not either full or empty, but there's a lot in between. Of course, this is again ignoring the glycocalyx and all the different things that are happening at the surface, or adsorbed protein, or adsorbed whatever, as you'll hear later. There have been some recent work which actually are using complementary methods to measure the refractive index of EVs, and we know that there's a spread of refractive indices, and yet we're still sort of assuming that median refractive index. So this is just a kind of, another wrinkle in all of this analysis. The lamellarity is, again, pretty much ignored in most literature, I would say. It's treated almost as like an artifact or something of interest when you do cryo-EM, and some people are even maybe blaming the different isolation methods for creating these structures. But as this sort of evidence mounts, and this is an awesome cryo-EM I found on Twitter, or whatever it's called now, where this looks like a clear evidence of alien EVs, ETs. Amazing. So of course, in synthetic materials and liposomes, these have been well known and studied for many years. There's ways to make multivesicular vesicles or multilamellar vesicles to hold more hydrophobic cargo, to do lots of interesting things for drug delivery and in different engineering applications. There was a fantastic paper that came out pretty recently from our next speaker, Ken Whitwer and his team, which actually proposed that perhaps that lamellarity is indicative of different biogenesis mechanisms, and that this is something, again, that's not an accident, but that is some way that the cell is preparing these. And I would say little is known about the mechanisms and also the implications. I even think in Ken's paper they referred to these as speculative mechanisms. So this is a great start, but there's a lot of work here to sort of understand what this means. And just more evidence from the literature. I mean, this is already many years since this paper came out. We've known about this problem and even started to quantify it, that the degree of lamellarity is not a small fraction of particles. It's many of the particles that we see. In fact, in my own lab, we've started to count thousands and thousands of particles from many different sources on cryo-EM to try to characterize this. And you can sort of see the total size distribution of all particles that we can find. In green here, you see how many and what size are the ones that are only circular and unilamellar, and they're small. They're all under 100 nanometer practically, invisible to most light scattering techniques. And in fact, what we're seeing, the highly scattering particles and also the larger ones are mostly multilamellar, or at least non-circular. So kind of an interesting trend, unpublished, so no reviewers have got their hooks into this yet, so it's probably not true, right? And this isn't an artifact of isolation methodology. So there's excellent evidence from recent literature that show you see these structures, especially non-circular vesicles, in a very high number percentage, so more than 50% in some cases, independent of which isolation method was used to isolate them. Another sort of myth that I'd like to dispel, or at least introduce some complexity, is the stability of these particles. There's this sort of, in my opinion, misconception that, one excellent control you can do is just throw a surfactant or a detergent at your EVs and the true vesicle should lyse. Everything else will remain there, and so this is a way that you can easily confirm that whatever mechanism or dye or whatever you're trying to measure is potentiated by vesicular particles instead of the non-vesicular extracellular particles. In the recent MySev, there's even a sort of statement that to confirm that fluorescence is associated specifically with EV, surfactants can be used to disrupt vesicles. They actually acknowledge that they may also disrupt certain NVEPs. But in reality, that's not the case, and it depends. Some surfactants, of course, can lyse particles. Some can lyse non-vesicular particles. It all depends on the surfactant that you use, the concentration that you use, and there's a lot of evidence in the field now that shows some of our most standard methods, like 0.5% Triton X100, actually do not lyse particles at all, and cryo-EM evidence of this show that these particles are still vesicular, and it doesn't just lyse the vesicular particles. And again, it depends on which surfactant and how much you use. It's worth noting that in many other fields in synthetic nanomaterials, detergents are used actually to make particles. So they're used to lyse cell membranes and form them into small particles and aggregates on purpose. And so it strikes me that this practice of throwing surfactant at particles, when not properly done with controls and analyzed probably by an imaging method, you might be creating new particles, in fact. A couple other strange things that we found, and I should have acknowledged that a lot of this was, I put out a call to the IOC and many people just to share with me weird studies and weird papers of how EVs aren't how we think of them as these typical cartoons. So I'm throwing a lot of these together. This is one from Rink and his group. They noted that when you isolate particles from blood, you're left with a supposedly insignificant number of platelets. This is less than 1,000 platelets. There's trillions of EVs in that same prep. These platelets are undetectable by most blood lab platelet counters, so it seems that you don't have any platelets left. Of course, flow cytometry can pick those off by their CD61 expression. Well, they calculated that the volume of these platelets, these 1,000 platelets, is more than the volume of the trillion EVs, and in fact carry an equal number of microRNAs to these EVs. So these tiny throwaway contaminant cells that you would think are very small in number can potentiate much of the function, or at least carry much of the biomarkers that we would commonly associate with EVs. I do a lot of work with blood EVs, and I never thought to look for platelets, and I'm just wondering how all of my papers are trash. Lipoprotein, of course, is shown with imaging to associate with EVs, so I think we even heard today, and certainly it's the way I was thinking about it for many years, that we have to purify EVs away from lipoprotein. Well, they stick, they associate with strong kinetics, so the fact of even thinking to isolate them away from each other may be a misnomer. And then, of course, the surface traffic of nucleic acids, as I mentioned before. So I like visualization, so I tried to put something together. I tried to cobble together the most accurate counts on average of blood concentration of red blood cells, platelets, EVs, lipoprotein and protein aggregates, and sort of colored them and made a little animation here. And this is a 60 micron section of a biofluid, and this is probably about the concentrations that we're thinking. So I don't know, for me it was interesting to think about, I mean, there's a lot of assumptions here, and I'm happy to talk about the details with people. But it's sort of interesting to see this staggering count. These numbers of EVs were actually taken from a new study that I have the pleasure to be a part of from Britta Betten and Edwin van der Poel and Rink's group, and they sent out many samples to a bunch of different labs and concurrently analyzed them by flow cytometry. And one of the pieces of data they were able to take out of this was actually to calculate the average concentration of EVs in a healthy individual's blood. So that's where this number 2 × 10⁷ comes in. I showed this to Rink and his team, and they were aghast because they said, well, wait, that's only above the detection limit of our worst flow cytometers. It's actually, the EV count is likely 10 or even 100 times more than that. I tried to simulate what 10 times more EVs would look like in that same image. I did 100 times 2, and it was just blue. It was only blue. Another interesting part of this is sort of considering the rheology of blood, that these are non-Newtonian fluids and that the particles and cells are not distributed homogeneously and probably are getting crowded towards the outside of the lumen of blood vessels. You can start thinking about what happens in microvesicles or micro arteries are, different. I'm not a biologist, just gonna put that out there. That the constriction of these may even exacerbate the concentration of EVs towards the endothelial barrier. So it's kind of interesting. We heard lots about circulation half-life today, so I won't belabor the point, but it's commonly measured to be much, much faster than your cursory EV introduction might have you believe. Some studies showing that it's even 2 minutes or shorter. It's surprising to hear from Charles and other folks that these dyes can persist for weeks or months, and thinking that we're measuring the probe after all, as Vincent nicely summarized. I really got scooped on this slide. I was so excited to burst everyone's bubble that there's hardly any evidence that EVs actually cross the intact blood-brain barrier. That was well motivated by our panel earlier. I want to believe it. I think it would be wonderful if EVs just crossed every barrier, but from what I can find, and I would love to be corrected and shown primary evidence in the literature, there are 2 studies that actually show direct evidence of EVs crossing the blood-brain barrier. One is the Murad ACS Nano paper that Vincent showed before, and the other was this other paper that I was able to find. This myth is so I wouldn't say it's a myth. I don't know if it's false. I just know it hasn't been proven true. It's so pervasive in the field that I found 5 reviews published in the last 6 months that claim that EVs cross the intact blood-brain barrier without citing it. So it may or may not be true. I think another complexity is the extracellular matrix. It's another sort of ignored aspect, that when EVs leave cells, they have to cross through these barriers which react dynamically and bind to EVs depending on their surface proteins, and that these might act as source and sink for EVs and completely change their biodistribution. And then finally, the concept of xenoparticles, which we asked everyone to consider today. Not really a different species, but what about nanoplastics? I don't know if anyone is following this scary modern news and literature, but nanoplastics are found in very high numbers in blood and other circulating biofluids. They scatter like EVs, actually more. They look like EVs. They can bind protein and albumin and other materials. So it may not be that these reference materials that we're thinking of just that we use to test out or spike into our EVs, but we actually might be directly measuring nanoplastics and other type of particles inside of our biofluids. You may not be surprised to know that there's many examples of nanoplastics crossing fetal and blood-brain barriers and doing many of the things that we ascribe to EVs. And it's sort of, I think the interaction of these two fields over the next years, because we share so many methods and complexities, will increase. But I thought it would be an interesting thing to bring up too. So how can we deal with all this information? I thought of a couple of things that maybe we can take away from this. The first is to not let our expectations of that an EV should be this beautiful single, bilayer particle to bias our data collection. So I think it's quite easy, for example, on a cryo-EM to go and search for what you perceive EVs to be. And I can't tell you how many papers that we've reviewed in my group or that I've read, which have figure 1A. It's a picture of a single beautiful vesicle from a cryo-EM image, and they say, we isolated EVs in high purity, and then they move on to human in vivo work or something crazy. So I think being aware of these complexities, actually embracing that EVs are rich and weird and different, and probably not that cartoonish picture that we imagine, actually is part of the pursuit I think we should be trying to go after. I also am a big fan of electron microscopy. I think actually viewing these things is very important. And then finally, start to introduce some of this flavor in your images. So we're starting to see lots of interesting images come out that show at least diversity in size, but think about lamellarity, density, surface materials, and other sort of weird things as you're starting to draw pretty pictures of these awesome particles. So okay, I went way over time. I'm going to turn it over to Ken, and Ken's coming from Johns Hopkins all the way in the US. I did a simple experiment where there's the thing called antipodes. You can actually look on the map of what is the opposite of Melbourne. It's pretty close to the East Coast in the US. So I think Ken came the farthest from anyone here at the meeting, just in terms of the physical distribution on the map.
Except for ET.
Thank you.